All right, what do you want to go over? Do you want to go through these and see if there's specific questions? Yeah? Okay, since we didn't say anything otherwise. Okay, so this is a unit 5-1. First topic up is meiosis. Okay, we've got some I can statements. I can explain the ways that meiosis results in the movement of chromosomes from one generation to the next. I can explain the ways to reduce the chromosome number of meiosis from one generation to the next. I can explain the phases of meiosis 1 and 2. I can explain the role of meiosis 1 and meiosis 2. Any of those you want to go over? We're not overly concerned about meiosis? Probably not, right? Not of the topics listed. Okay. Mitosis. Mitosis is not in this unit. However, they could ask you to compare meiosis to mitosis. That is a possibility. It's more understanding that like mitosis makes body cells, meiosis makes sex cells, mitosis makes identical cells, meiosis makes non-identical cells, mitosis goes diploid to diploid, meiosis goes diploid to haploid. It's stuff like that. Back when we did meiosis, you remember we compared them. Do you understand those comparisons? Do you understand the places mitosis would be used versus meiosis would be used? Okay. All right, then we get into genetic diversity. Can you explain the ways that meiosis generates genetic diversity? There's 3 ways. What were they? Independent assortment, random fertilization, and crossing over. Very good. Can you explain how the homologous chromosomes separate during anaphase? I can explain how the daughter cells receive both maternal and paternal chromosomes. I can explain why the daughter cells after meiosis 1 are haploid. I know what crossing over is. I can explain how crossing over increases genetic diversity. I know what independent assortment is. I know how independent assortment increases genetic diversity. I can explain how random fertilization increases genetic diversity. Questions on those? Still feel okay about? Yes.
What's independent assortment?
Okay. Independent assortment is the fact that our tetrads, when our tetrads line up down the metaphase plate, nothing dictates if they line up this way or this way. Okay? So in every tetrad, you have chromosome number 12 from mom, copy of chromosome 12 maternal, chromosome 12 paternal, copy of chromosome 12 paternal. So it basically, independent assortment says it doesn't matter if your maternal or paternal chromosomes line up on the left or right. Depending on how those go, you could theoretically create a gamete that had 100% maternal chromosomes, 100% paternal chromosomes, or some variety. You figure it out by doing 2 raised to the power of tetrads. So for humans, what is that? 2 to the 23rd. Okay, that tells you how many possible ways independent assortment can line them up. Good? Mendelian genetics. I can explain ways shared processes support the concept of common ancestry. I can explain ways shared features support concept of common ancestry. I can explain ways a conserved process supports common ancestry. I can explain, okay, that gets repetitive. I can explain ways fundamental processes support the concept of common ancestry. I can explain ways, okay. I can identify carriers of genetic information. I can describe the cellular location of ribosomes. I can describe the ways that the genetic code demonstrates common ancestry. I can identify the metabolic pathways that have been conserved across all domains. All right, do you have any questions about those? Do you get what it's getting at?
Okay, what are the metabolic pathways that we use now?
So like cellular respiration, portions of cellular respiration are the same, like glycolysis is the same across all organisms, stuff like that. Okay, you don't need to have specific examples, but so anything that's considered highly conserved, anything that supports common ancestry just means it's something that happens the same way across a vast majority of organisms, indicating it evolved a long time ago, and then it's just been passed along. We talked about that with like the central dogma, transcription and translation being the same in all living things, kind of regardless of what organism type they were. Anything else from that? That's just getting a little bit, we get into all of this in more detail when we do evolution. Some of these topics that they give in College Board do kind of cross multiple units, and this is one of them. Yes, you need to recognize that some of the stuff we talk about in genetics does lead to how they support evolution, but We'll talk about it a lot more then. I can explain the law of independent assortment. I can explain the law of segregation. I can identify the expected ratios for a monohybrid or dihybrid cross. I can identify using quantitative analysis whether the observed phenotype is statistically significant. Feeling there's questions there. Yes?
An example of a polyhybrid cross.
A what hybrid cross? Oh, yeah. Sorry, poly is the word you said. Sure. Okay. That's not for you. So like one of these. Okay, tell me the probability of having an offspring that's entirely dominant. Offspring that's entirely dominant. Tell me the odds. Go. I'll give you guys a 15-second lead and then I'll start writing up the answer. What are the odds that an offspring is entirely dominant?
You wrote F, you make that a back F.
Oh, I wrote FE. I got distracted at the end apparently. That's a mutation. Thank you.
Stafford, phenotype or genotype?
Phenotype. Good question.
Wait, what is it that's all dominant?
Dominant phenotype.
Oh, okay.
Yeah.
All dominant? Okay.
So like the traits that have to do that.
Try and do this without drawing any Punnett squares. Try and picture them in your head. For time's sake. Say it again. Thank you. I did not want to do that much mathematics. You two get the same thing. Does everyone understand how I got that? Yes? No? Maybe? Question? Sure. So, remember, we assume law of independent assortment unless we're told otherwise, which means the inheritance of one gene in no way impacts the inheritance of the next. So I can look at these as individual Punnett squares. And then you're going to want to, ideally, just picture the Punnett squares in your head, not actually draw them. You can draw them if you need to, but that's going to be rough on time. Okay? So I know when I cross a heterozygote and a heterozygote, the odds of getting a dominant phenotype is 3 out of 4. So I can do that for any of my heterozygote ones. A, C, D, F. Okay? Then for B, though, I have a homozygous dominant with a heterozygous. Well, that's a 100% chance that the offspring are going to be dominant phenotype, so I just put a 1. I take all those numbers. I want to know what the odds that these individual events are going to happen at the same time, so I multiply them out. There's an 81 out of 256 chance that these babies will be dominant phenotypes. Questions? Do you need another one or yes?
How if you do this if it was a linked gene?
Okay, great question. If it's a linked gene, you're not doing this. Okay, so where could that come up though? There could theoretically be a question where they ask you to do this, and then they tell you how many actual babies they got that were fully dominant, and say it doesn't match. And then they have you run chi-squared. And then you say there is a statistically significant difference between what you predicted and what you got. And then they could ask you, how could that be? And then you could say, oh, maybe one or more of the genes are linked and it messed up our predictive ratio. That's what you could do. Okay? Yes?
Can you give an example of a chi-squared problem?
Yeah, sure. Trying to think if it's gonna be easier. Okay, do you guys have your formula sheets? 'Cause you're gonna need them for the, I mean, not for the test, I'll provide them, but you need them for that chart, the probability chart. Dig that out while I think of a problem. Don't forget your calculators tomorrow and
We can use a
Thursday.
calculator on the whole AP test, right?
yes, you can, and mine.
If you know how to do chi-square calculator, we can do that.
Correct? Nobody can stop you. I'm gonna pull one because if I do the numbers wrong, I won't know off the top of my head how it's gonna come out. Give me 1 second.
That 107.
Where?
I don't know. I don't know. Was that W7? With?
With.
Oh.
With. How was your test?
Fantastic. Wait, is nothing just not here yet?
What?
No, it's been over.
Isn't a lot of these people math having tests? Like, yeah. Okay. You need to determine the inheritance pattern. Based on the inheritance pattern, you need to predict the expected ratios, and then you need to determine if the offspring match the expected ratios or not using chi-squared analysis. It should take you about 5 minutes. I said so many words. Okay, so try that again. You need to figure out the inheritance pattern. Based on that, you need to figure out the predicted offspring. You need to do the predicted offspring versus the observed offspring and use chi-squared analysis to determine if the differences between them are by chance or due to incorrect inheritance pattern. It's a 2, I'll fix it, I'm sorry. It's a 2. My handwriting's tired.
Are we figuring out the inheritance pattern based on the numbers you gave us?
And what you know. The combo. But yeah.
drop protection.
Broken?
Yeah.
Not broken? Yes, broken.
Not sure.
Okay, let's hope with not broken. Seems fine? Good. If it was closed, they usually do okay when they fall.
So, we know that colorblind is like recessive. So to figure out what the probability should be, so if you want to know the variation, that would like x lowercase a. So.
Write it down.
Yeah, so what do we do with it?
You go to your table. So that is called the transfer to the table. So we know as one is the right.
I'm ready. I'll take it.
We don't know what this one is. Could be. So if this makes, if these two things, right, this is x and minus 1, so what about 3? Either way, this is, for females, this is, I always like, I know they don't have a word. So, we know one of the mom and one is not fully, she's not covered. Right, so if this happens, the child is fine. If this happens, the child is fine, right? So we know these two are definitely recessive. So we don't know if this is X capital or recessive or dominant. So if it's recessive, then either of these, the child is either of these, the child would be right? But if it's dominant, then there's no chance of the child being recovered. So anyway, you're so this can be. Oh wait. Do write down. So half of them here.
I think I'm about to get it.
Are half of them here are copies. Okay. So the other option is I'm just gonna write. In other words, if the mom is dominant, everything is just happening. Right? So in how many of these? In a quarter of them, the child's covered. Okay. So and does it depend on the child?
This is the same one. Oh, well, so this is 0.7.
Right. In this case, it really doesn't count.
So basically, since this
in every case,
value
if
is
the
less than this,
female colorblind, if the female's colorblind, the male's
if it's
colorblind.
less than, because remember in chi-squared, the highest value is more.
Because if this was a capital B, no matter what happens, if it was dominant, she's
It would have been not.
homozygous
That means
dominant.
it doesn't.
This kid will be either hemizygous dominant or fully dominant. Whereas if she's whatever here, which is heterozygous, there's a 50% chance that the kids are not, and a 30% chance that they're not. Okay? So
Sean, did you get this number? Yeah, so that's,
Then, I mean, it's not.
it's not significant.
All right, but like this is, yeah. I think I did it right then. I'm good.
But she told us that it's
Right, that's what I
it's one of the.
I think I did it.
You just.
Yeah, that's what I was thinking, but then it like the numbers were off. So
Yeah,
it's like,
but it doesn't always have to know stuff because, like, in a case like this could very easily, if it isn't any generic text link break, it still shows that this math is not incorrect. It's still like. It doesn't always mean that. So there's a 25% chance that the kids come off as come off as part right? So now we're trying to figure out what this.
Let's go through this one together. We're hitting a point where everybody's stuck.
Get points then.
All right. So
I didn't do any calculation.
you've got to commit to a hypothesis first. What are our possible hypotheses?
Normal inheritance.
Standard Mendelian inheritance with what being dominant?
Non-colorblind.
Non-color blindness, or
Sex.
sex-linked inheritance with it being recessively inherited color blindness. You gotta pick one and stick with it. You can run either. It's a hypothesis. You can run either. But your math has to match your hypothesis. Which one do you want to run first? Normal. Okay, sorry, you lost that. If we run normal, does gender matter? No. So we need normal and colorblind in terms of our offspring. So you need to take 126 and 132, right? And what is it, 160? What? Oh, there should be 2 there, huh? Okay, we need our total numbers. We need to figure out what the genotypes of the parents would be if it's inherited with standard Mendelian genetics. Dad has it, so dad would have to be homozygous recessive. Okay. And the female? Because they have kids that have it and kids that don't, and she doesn't. Okay. If these are the parents, what ratio of offspring should we get that have it and don't have it? 50-50. So what's that work out to as our expected values? 259. Is that what you said? Observed minus expected squared over expected for normal. Observed minus expected squared. You got what I was trying to say? Squared of colorblind. Sum those together, and what number do you get? Can you give me it as a decimal, please? As on both other ones, because we have to put it on the chart.
0.0077.
Can someone confirm this number? Did anyone else run this chi-squared? Can someone confirm this number? Did anyone do the math?
I just did it right now.
Is it right?
I think it's right.
Okay.
I don't see how it's wrong.
Okay, that's fine. I just didn't do the math. Does it look good to everybody else? I can't confirm it. I don't have a calculator right in front of me. Observed minus expected. Right. Okay. What is our degrees of freedom for this one? 1. So what is our cutoff value at 0.05? Who has the chart? 3.84. 3.84 is our cutoff value. Is there a statistically significant difference in the data? No. That's if we assume it is standard Mendelian inheritance. What if you ran it as sex-linked? Okay. Then, what must dad's genotype be?
Hemizygous recessive.
Hemizygous recessive. Mom.
Heterozygous.
Heterozygous still. What types of offspring do we get? Like ratio. What's the ratio of offspring? 1 to 1 to 1 to 1, males to females and colorblind to not. Okay? So these would be our observed values. What would be our expected values?
129.
For each one. So then you have to do observed minus expected over expected squared 4 separate times and sum them up. What do you get as your chi-squared value?
0.703.
That one?
Yeah.
Okay. What is our degrees of freedom? 3. What's our cutoff value?
7.82.
7 point what?
81.
In both cases, are we finding a statistically significant difference?
No.
In this case, can we actually dictate what inheritance pattern it is from this data? So both of these would be completely accepted answers if this was the exact problem you were given. Okay? The biggest issue I could see happening, your math has to match. If you're claiming it's not sex-linked, don't run your numbers like it's sex-linked. Sum them. If you're claiming it is sex-linked, run your numbers like it is sex-linked. In this case, with this particular scenario, we don't have a way to tell. Yes?
Could there be a situation where, like, if it was sex linked, it would be statistically significant, but
Yes. Yes, that's a possibility.
So like, would we just say like fails and
Correct. In both cases, there's not a statistically significant difference in the data, in the observed and expected data. What else? What other questions on this? Yes.
The degrees of freedom is just the total number
Minus 1. What else can I answer? Otherwise, we got to move on. Yes.
So, for this type of problem, it does not matter if we're right about it being sex.
In this exact instance, no. This is the problem of pulling practice problems instantaneously and not fully being able to do all the math for them. In reality, if you have this on a test, there'll be one way that it has to work.
Can you think of an example of
Well, like we did the practice one a while back with the fruit flies, remember? Okay. It was different eye color and fruit flies, and it only, like, in that case, it was obvious because males were all one eye color and females were all the other that it had to be sex-linked. So usually it will only follow one pattern. This is just a bad case of it working out this way.
So for that fruit fly problem, had you done,
The other way, you would have rejected really badly.
and that was putting marking correctly, or that? Because you
Yes,
still did
because
that.
they were testing you on being able to determine the inheritance pattern. You'd probably still get the chi-squared points
Okay,
though,
okay.
but you get the inheritance pattern wrong.
That makes sense.
What else? Yes. If it's on the table at any way or greater, you reject. Because this table only shows the rejections. The way that they give you the table, it only shows you the reject values. You want to keep going with this? Yes, no? Yes. What's the ratios, expected ratios in a monohybrid cross for phenotypes? Phenotypes? 3 to 1. Genotypes? No. We're still on monohybrid. 1 to 2 to 1. What's the phenotypic ratio for a dihybrid? 9, 3, 3, 1. Make sure you know those. Non-Mendelian. Can you explain what linked genes are? Can you determine the distance between two genes on a chromosome using recombination frequency? Can you determine the inheritance of a sex-linked trait based on a pedigree? You can look at a pedigree and figure out if it's sex-linked.
Wait, a second. What are the monohybrid and dihybrid ratios?
3 to 1, 9 to 3 to 3 to 1. Yes. Sure. Recombination frequency. Recombinance is a fancy way of saying they don't match the parent phenotypes. Okay? So if in the made-up world the parent phenotypes are one parent's black and brown and the other parent's red and yellow, recombinants would be ones that are like black and red, or yellow and brown. They're a combo of the parents. They're a recombinant. So you calculate recombination frequency by adding up all of the offspring that don't exactly match either parent, and you divide it by the total number of offspring. That decimal point equals your distance in map units that the two genes are apart. It also tells you the recombination frequency, or how often crossing over happened. Because the only way you got those offspring is via crossing over occurring. Okay, so like, let me pull that to show you one. Let me try and find numbers. Give me 1 second. For genotypes, monohybrid cross.
So it's 1 to
It genotypes. Phenotypes is 3 to 1.
Wait, is that for dihybrid?
No, monohybrid.
So monohybrid is 3 to 1 and then phenotype
Yes. 1 to 2 to 1.
is just like for monohybrid it's 2 phenotype to 1 phenotype. What about for dihybrid?
I'm sorry, I'm trying to read. What? We don't know genotypes for dihybrid. We don't memorize that because there's 64 of them. All right, let's look at this. While working with a type of beetle that's normally smooth, large, and white, you discover 3 mutations that lead to the recessive phenotypes bumpy, small, and gray, respectively. You cross true breeding smooth white beetles to true breeding bumpy gray beetles and get 100% smooth white beetles in F1. What's that tell you about smooth and white? Dominant. You then cross the F1 beetles to a true breeding bumpy gray beetle. So you're crossing it to a homozygous recessive, homozygous recessive. And after analyzing 800 F2s, you calculate a map distance of 5 map units between the smooth and color loci. What does that mean? 5 map units. They're linked. They're only 5 map units apart, so crossing over is only going to happen 5% of the time. That's what that would mean. Okay? That's what that would mean. Does that make sense? How would we have gotten it when we're looking through those 800? You would have expected a 1 to 1 to 1 to 1 ratio of offspring. You would have expected 25%, okay, to be smooth and gray, 25% to be smooth and white, 25% to be bumpy, and you see what I'm saying? But you won't get that. You'll get most of them being smooth and white or gray and bumpy, and only 5% were not one of those 2 things. Okay? Okay. I can determine inheritance of a sex-linked trait based on data with a parental phenotypes. So this is all about sex-linked. You don't have to do polygenic. Ignore that. Sometimes it includes information you don't need. I can identify organelles other than the nucleus that contain DNA. Do you remember that? Mitochondria and chloroplast. We don't talk about the chloroplast as much because nobody likes plants that much, but mitochondria have their own DNA, so you technically can track inheritance via the mitochondria. The thing is, mitochondria comes from who? Your mom. So it only lets you track maternal lineage, basically, is what you can do with mitochondria. It's not genetics in the same way, but you can track your maternal lineage via mitochondrial DNA. I can explain the ways the same genotype can result in multiple phenotypes under different environmental conditions. That could be like the Arctic fox, coat color being different summer to winter. That could be things like tanning, okay, under like, well, those of us that can and can't, right, tanning under certain sunlight. That can be getting into epigenetics, whether the genes turned on or turned off by certain environmental conditions. I can describe phenotypic plasticity. That is a fancy way of saying the phenotype can be changed based on the environment. That's like the fact that the fox's coat color changes. Or do any of you have hydrangeas in your yard? What color are they? Blue, purple, does anybody have pink? It's based on the pH of your soil, what color they are. You can change the color of your hydrangeas by changing the color of the changing the pH of the soil right underneath them. That's phenotypic plasticity. Okay. I can describe ways environmental factors influence gene expression. That could be like the lac operon. Okay. They can't make any of the lactose enzymes unless lactose is present. Right. That's an environmental impact on gene expression. That could also be anything we talked about with epigenetics that turns certain genes on and off based on like calorie intake, stuff like that. I can explain ways chromosomal inheritance generates genetic variation in sexual reproduction. We've talked about that. That's independent assortment. I can describe segregation and meiosis. We talked about that. Independent assortment. You know what fertilization is. We know how they lead to genetic variation. You know how genetic disorders result from a single affected allele, right? That's getting into protein synthesis. How could one allele end up with a different shaped protein, which means it has a different impact? I can describe genetic disorders that result from chromosomal changes. That's like non-disjunctions. That's like Down syndrome, stuff like that. I can describe a non-disjunction. That's during anaphase 1 or anaphase 2 of meiosis. Either the homologous chromosomes don't separate or the sister chromatids don't separate. And you end up with one too many or one too few chromosomes in the resulting gamete. And then that gamete becomes a zygote that now has one too many or one too few chromosomes. Right? Down syndrome, again, is the most common example. I can predict the results of a non-disjunction. If they showed you a picture, could you explain, oh, that gamete has one too many? Oh, that gamete has one too few. Could you explain that? Okay. I can describe the structures involved in passing hereditary information from one generation to the next. That's DNA replication, that's understanding meiosis, et cetera, et cetera. I can identify DNA as the primary source of heritable information. I hope so. I can describe the way genetic information is transmitted from one to the next, same thing. I can describe how DNA is stored. That's talking about chromatin, heterochromatin, euchromatin. That's talking about the histones that DNA wraps around. That is talking about the ways they wrap around it. I can identify different genes, prokaryotic and eukaryotic genome shapes. Prokaryotes, it's what shape? Circle. Eukaryote? What do they call ours? Linear. I can identify differences between amount of prokaryotic and eukaryotic genome. Who's bigger? Who wins? We do. What's a plasmid? What do they call it? Extra nuclear DNA. Who has plasmids? Only bacteria. Extra little pieces that have 1 to 3 genes on them that are not technically necessary for life. Humans use them a ton in biotech. Why did your stuff glow today? Because we put a plasmid into the E. coli that contained a glowing gene. I can describe biotechnology used for plasmids. The lab we just did shows up on the test every other year, basically. It shows up often. It's a lab that they love to ask questions about. They don't expect you to recall info, but they could give you basically the same scenario you did and ask you information about why did these glow? Why did these not glow? Why was there no growth on this plate? Why was there growth on this plate? How did you make them glow? Et cetera, et cetera. You know what DNA looks like? I hope so by this point. You know how it's hereditary material? I hope so by this point. You know how the nucleotides base pair. If you don't know that at this point, just go home, okay? Purine, angels. Angels are pure. A and G, they have the 2 carbon rings. Pyrimidines, they all have Ys. Pyrimidine, thymine, cytosine, all have Ys. They have 1 ring. I can determine the number of other nucleotides. So could they give you a practice problem where they ask you for the percentage of cytosine by telling you that the organism has 12% adenine? Somebody do that out loud for me. Be loud and do the steps.
If there's 12% adenine, I mean there's 12% thymine, which means the rest of it is.
So 12 and 12 is
24%.
24%.
Means the guanine and cytosine
Must be
76%.
and divide that by.
Is 38.
Yeah. Good job. Did everybody get that?
Wait,
Yeah.
so what are the 2 functions of plasmids?
Where are you?
Actually,
2 functions of plasmids give like antibiotic resistance, give a gene that they pick up a gene that the organism does not currently have, and then that we use them in biotech. We use them to purposely. They make insulin for humans using what we did with the glowing. They cut the insulin gene out of a human, they put it on a plasmid, they put it into vats after vats after vats of E. coli, they let them replicate, they let the E. coli start pumping out insulin because they start making the protein out, we extract it out, put it in a vial, that's what they give you if you're a type 1 diabetic. It's all based on the same thing you did here with glowing, that instead of glowing, it makes insulin. I know how genetic information is copied, DNA replication. I can describe the directionality. That's the anti-parallel, the 5' 3' DNA polymerase sucking at its job. I can explain helicase breaks the hydrogen bonds in the parent strand. I know topoisomerase prevents supercoiling. I know that RNA primers are necessary because DNA polymerase 3 can only add to existing nucleotides. I know primase or RNA polymerase puts down those primers. It has directionality, but it does not have the issue of adding to nothing. I know leading and lagging strand is based on the directionality of the parent and the directionality of the helicase on the replication fork. I know ligase builds the covalent bonds between the sugar and phosphate on Okazaki fragments. Is there any of that you didn't know?
I have a question.
Yes.
So, aren't like RNA primers and primers the same thing? Or like,
Primase and RNA polymerase are the same thing.
okay.
Primase is like a nickname for RNA polymerase. Yes.
I have a question about the experiments.
Sure.
What's the difference between the Avery and Chase experiment and the Griffith experiment?
Let's see. Avery and Chase did the virus. No, they all did the mice.
Yeah.
Yeah? Griffith repeated it, but later.
do we need to know this?
No.
Okay.
Griffith just repeated it. It's the same experiment. Okay, you understand what transcription is. You know the central dogmas. Transcription, translation is the same in all organisms. You know mRNA is the messenger. tRNA brings the correct amino acid. rRNA is what makes up the ribosome, which acts as a big enzyme or a ribozyme to put everything together. You know how mRNA, tRNA, and rRNA come together in translation in the cytoplasm. You know that the DNA sequence becomes RNA sequence based on complementary base pairing. You know RNA polymerase puts on the complementary base pairs. The template strand of DNA is the one that, okay, this confuses people. The template strand of DNA is not the one we make RNA out of. You make RNA out of the non-template strand. Okay, there's 2 strands of DNA. They call one the template, one the non-template. It opens up, RNA copies one of them, right? RNA copies the non-template strand. Okay, everybody take a second. Why does that actually make sense? That we make the complement of the non-template strand, not the template strand. Everybody think for just 2 seconds before I let anybody say it out loud. Okay, nice and loud.
Because then when you put it back together, you get the original.
Right. by making the complement of the non-template strand, you're technically dead copying the template strand. Because remember, mRNA is not actually a copy. Where there's an A, mRNA puts a U. Where there's a C, mRNA puts a G. So it makes the complement not actually a copy. But that means you end up making an actual copy of the template strand. Did everybody gather that? I can identify the non-coding strands. Okay, you don't need to do minus and okay. So what it is, they're throwing up any vocabulary word they've ever seen on an AP test. They have not used antisense or minus in like a decade. I would not worry about that. They currently use template and non-template. And half the time they don't even use that. So I wouldn't worry about it that much. I understand the directionality of transcription. Remember, there's still a leading strand, lagging strand issue, but since you're only copying one, it's not as big of a deal. I can describe post-transcriptional modifications. What type of organisms can do post-transcriptional modifications? Only eukaryotes. 5' tail, poly A cap, introns remove, exons spliced together. You know the function of the poly A tail and the cap? Threefold. Help it get out of the nucleus, help it trap the ribosome once they're there, help prevent destruction of the mRNA in the cytoplasm. You know what RNA splicing is? Take out the introns, put together the exons. Alternative splicing dictates basically different ways to put the exons together to get you a different product. That's how humans can only have about 30,000 genes, but can produce about 100,000 protein products.
Wait, the three functions of the polypeptide gene and the cap are to get out of the
Nucleus?
protect the stuff, and then connect to the
Ribosome. Yep. All right, I can describe the ways phenotype of an organism is determined by phenotype. I think that means by the protein is what they're trying to say. Like how the protein equals phenotype. There's a typo in there. I can identify the location of translation. It always starts where? Translation always starts where? Cytoplasm, but sometimes ends where? Endoplasmic reticulum. If the protein needs to be exported out. I can describe the differences between location in prokaryotes and eukaryotes. Big difference is prokaryotes do not have a nucleus, so transcription and translation occur simultaneously in the cytoplasm. You know what translation is, right? Turning mRNA language into amino acid language. You know how we elongate, you know we terminate. There are 3 specific stop codons that do not code for a tRNA, but instead code for a protein that breaks apart the ribosome complex. Translation starts in the cytoplasm in the P site of the ribosome on the start codon. You know what a codon is, 3 nucleotides per 1 amino acid. You could, if given the table, figure out the codon to amino acids, like you can use the table. You understand that there are 64 possible ways to put the 4 letters together into a set of 3, but there's only 20 amino acids, so you're going to have overlap. That's how you can get silent mutations. That's how you can change the DNA, but get the exact same protein product. You understand that we all use the same genetic codes that also does common ancestry, and we know for a fact it's completely the same because you guys just took a gene that they took out of a jellyfish and made bacteria glow in the dark. Theoretically, they could do that to us. You can mix and match because it's all the same. We're making different products out of it, but it's all the same sequence of letters. Ribosome has an A, T, and E site. A and P are where the tRNAs come in, and we keep adding new ones to the A site and keep growing the chain. E is for exit. Okay, what is a retrovirus? Viruses are not on this test. They're not. Viruses are not in our curriculum. They're not. That being said, they like to use HIV as an example a lot. Like, you know how they give you a ton of background reading? HIV is a common thing they use. So I just wanted to take 2 seconds to talk about it. HIV is a retrovirus. What's unique about it, what made HIV so able to infect, right? And so complicated to figure out. HIV, just like every other virus, has a nucleic acid portion and a protein portion. The HIV nucleic acid portion is made out of RNA. Okay, some viruses are. That's normal. What's unique? HIV comes with its own enzyme called reverse transcriptase. And what it does is it does transcription in reverse. What does that mean? Turns RNA into DNA. So when the virus gets inside of your cells, that enzyme turns the virus's RNA into DNA, and then that DNA can join into your genome and become part of you. And it can hide out inside of you. And that's part of the difficulty that comes with these viruses. You don't need to memorize it, but you could see it as an example organism, so just be aware. Because they took viruses out of the curriculum. All right, you know how we regulate gene expression. The big thing is knowing prokaryotes versus eukaryotes, understanding operons and prokaryotes. Okay, you guys just had an example of an inducible operon. The GFP gene was not produced until that operon was induced. It didn't get induced until we had arabinose. Arabinose had to bind to the repressor, change its shape, make the repressor leave, and then we could transcribe and translate the glowing protein, and that's why after 24 hours, they glowed. That's why they didn't glow initially.
What is central dogma?
Transcription to translation. DNA to RNA to protein. That's the central dogma of life because all living things do that process the same way. Yes? Sure. So you can, if you methylate DNA by changing the structure of the DNA, it winds more tightly around the histones, which turns it into heterochromatin, which basically turns the DNA off because you can't transcribe it anymore. Yes? Repressible. That's one that's normally on, and normally the repressor does not fit anything, but if something binds to the repressor and changes its shape, it could go up and fit and repress the operon. Repressible is pretty much always on, but you can occasionally turn it off. Inducible is always off, but you can occasionally turn it on. Okay. With eukaryotes, it's understanding activators and enhancers, understanding that you have to have the correct activator proteins to attach to the enhancer regions or transcription will not start. That is the fact that a liver cell will never have the correct activators to make like a protein for your lung. Okay? That way a liver cell never accidentally makes proteins for the lungs because it can't. It's all control to make sure your individual cells stay the specialty they're supposed to be. If you're supposed to be a lung, you can only make stuff to be a lung. If you're a heart, you can only make stuff to be a heart. We need that level of control. What would it be like to have 100 trillion cells and have no way to ensure they only did the job they were supposed to do? That would be a disaster. We wouldn't exist. We wouldn't have evolved to be here if we didn't have control over that. Okay? Transcription factors are the end of the cell signaling pathway. That's the thing you turn on at the very end of the cell signaling pathway that starts up transcription. Okay, is this the last section or is there more? Oh, there's more. So prokaryotes are operons, eukaryotes use heterochromatin and euchromatin. Eukaryotes also use activators and enhancers. Eukaryotes also can break down mRNA in the cytoplasm using non-coding RNAs. Eukaryotes can also break down existing proteins using a proteasome, a protein that breaks down proteins, right? We can control transcription and translation at every level. We can control whether it starts. Once it starts, we can dictate how long the mRNA is around for. Once we make a protein, we can dictate how long the protein's around for. We control it at every level.
What about prokaryotes?
Prokaryotes just have operons. That's the only way they control it. Yes, both prokaryotes and eukaryotes have promoters. The promoter in a eukaryote is specifically called the tetabox. In prokaryotes, there isn't something specific like that, but it's a sequence of nucleotides that act as a substrate for transcription factor enzymes. Okay, you know what a promoter is. We just talked about that. We just talked about transcription factors. You know what RNA polymerase is. Okay, we're about out of time. Is there anything specific I can go over? Yeah.
How important is it to know like the difference between eukaryotes and prokaryotes?
You definitely need to, because if it's an essay question and you don't know which group it applies to and you say it wrong, you're not going to get the point.
So eukaryotes control, like they have enhancers, they can use the operons, they can do what?
No, eukaryotes don't have operons. Only prokaryotes do.
Sorry, wait, so what do eukaryotes have?
Enhancers and activators, heterochromatin and euchromatin, the ability to break down mRNA, and the ability to break down proteins. What else can I answer?
So really back to kind of what I asked, well, like the difference between RNA primers and kinase in DNA replication, or like.
Our primase makes the primers. Primase is the enzyme that builds the primers.
Okay. Okay.
Did that help?
And then the primers are the things that are actually like being laid down.
Right.
Okay.
Right. Yes.
So what are the people we're supposed to know?
People you need to know were Watson, Crick, Rosalind Franklin, Watson, Chargaff, Hershey and Chase. I think that's, no, and Methylson and Stahl.
What would an FRQ on this look like? Oh, that
Wouldn't be an FRQ, I've only seen it pop up in multiple choice. Can you be super loud, Nathan? I didn't catch that.
Found out it was one polypeptide, one protein.
Oh, we don't have to know that name. I don't remember it off the top of my head at the moment, but we don't have to know it anyway. What else? You got 90 seconds. Okay.
Do we need to know how like our RNA and tRNA are made?
You don't need to know how they're made. You need to understand that they basically act as a ribozyme, an enzyme that's made out of folded up RNA, but you don't need to get into how they're literally produced. That's beyond our scope. Good question. Yeah. It's how many times you do observe minus expected squared over that minus 1. How do you get degrees of freedom? What else? Yes.
If you were to reject a null hypothesis, can we like go over how to interpret that?
So it depends on what they're asking you to do, right? You could be rejecting a null, you could be rejecting an actual hypothesis. But basically what it is, if the number is less than what's on the chart, we're saying there's no statistically significant difference between your numbers. So your observed and your expected are the same for statistical purposes. If your number is greater than or equal to what's on the chart, we're saying there is a statistically significant difference. So your expected values are not correct. Does that help?
Yeah.
Okay.
It's like for the null hypothesis, it's just like
That there's
negative. Like,
one is not influencing the other. There's no impact. Like the independent variable has no impact on the dependent variable.
and so like I'm using like hidden activity, so it's just like the null hypothesis means that they don't have the impact.
Right.
And so then if there was no statistical difference, like saying that